the introduction. So the enormous advances in genotyping technology and the drive down of the cost per genotyping have resulted in an explosion of positive whole genomic association studies and the identification of many new genes for common diseases. So just around 1990s, researchers could already examine an individual's genotype only a single gene at a time, and this was worthwhile only in some few cases where the disease-causing mutation was already identified. And by the turn of 21st century, advances in genotyping already allowed scientists to look um, at a much larger sample of many nucleotide changes that differentiate one person's genomes from another. And so now, just in 2013, scientists had already the ability to genotype not just one or two or thousands of loci in a person's genome, but nearly all of the 6 billion nucleotides in a person's diploid genome alone. So, what is genotyping? So, genotyping is the process of determining differences sa genetic makeup of an genetic makeup or the genotype of an individual by examining the individual's DNA sequence using biological assays and comparing it to another individual sequence or a reference sequence. So, ang kaning genotyping, it uses sequencing information para to determine the genetic differences or ang variance sa individual or sa biological population. So kani siya nga type of technique, it used to investigate it is used to investigate predetermined and a very specific region of the genome. So for example, when thinking about human DNA, a high percentage of it is identical from one individual to another. So like more than 99%. But there is still variation from one person to another, that 0.1%, which makes us very unique. So genotyping can be used to analyze some of those differences. So in doing this, scientists look for information or the genetic variants, which occurs in several types. So muna siya, genetic variants, the SNP, the DIP, the or indel, the SSR o ang CNV. So this could be the genetic markers or kanang genetic variants that will be used by the scientists to look for the information.
Okay. Miss Anna,
So
sorry to interrupt.
yes,
That
ma'am.
is read as SNPs.
Snip
SNP.
was snips, ma'am. Sorry.
Yes, okay.
Snip. Oo.
That's okay.
So, so kani siya mo tong uh, these are used to target a specific position or the segment in an individual's DNA. So kani siya mo nang ang mga scientists they use this to as they ay more bag i-predetermine na nila nga kani siya nga mga genetic variants. This is where the information or relevant information that they will use in order to um sani? To know the differences between or genetic differences between individuals.
Okay,
So
Miss Almas, before you continue,
yes, ma'am.
so uh, if they find differences in the genetic makeup of individuals belonging to the same species, what does it indicate, or how are they going to use this discovery? Say, for example, we have races, diba, human races, and
Mhm.
then, okay, can you relate it to COVID, for example?
They would use it, ma'am, in order to identify unsa ang differences from one person to person. Then some na po ni siya uses, ma'am, for so like as a medicine kanang
Mm-hmm. So medicine. How about medicine? What about medicine?
it will give
Or what does this variance indicate?
this variant.
Maybe we can use this for, say, for example, we'll start first with evolution. Can you relate it to evolution?
Yes, ma'am.
Systematics and medicine.
Um, na to siya na siya sa importance na butang mo.
Sige, sige, sige. Go on sa. Sige, go on sa.
So, no, yun ako dito. So, ang genotyping can be performed through a variety of different methods depending sa variance of interest o sa resources na available. So, there, what's that? When it was. Ano ba ni? Ay, so there ay. So these are some of the current genotyping methods, which includes the RFLPI, the restriction fragment length polymorphism, identification of genomic DNA, the random amplified polymorphic detection of genomic DNA, the AFLPD, or the amplified fragment length polymorphism detection, the PCR polymerase chain reaction, then DNA sequencing, ASO or allele specific oligonucleotide probes, hybridization to DNA microarrays or bead. So can you show all of these are current methods, genotyping methods. Yeah, current genotyping methods.
Okay,
As
let
I've,
me interrupt.
sige, sorry ma'am.
So, kaning genotyping is actually one of the results of the different methods used, except for CRISPR-Cas9. Okay. So, for
Yes
example,
ma'am.
NGS, HTS, DNA microarrays, also known as DNA microchip, can be used in order to detect genetic polymorphism among individuals of the same species. So, genotyping is an application to all of the methods that I have assigned to you. So katong na ay NGS, HTS. Those are just few. So some of it also includes the one that is um, sani? Kanang listed on the presentation of
Those
Miss
are
Elmas. Okay.
okay.
So those are examples of DNA
Application
sequencing method. Thank
method.
you. Go on, Miss
So
Elmas.
due to current technological limitations, almost all of these methods or the genotyping is partial. So, as I said nga, that is only a small fraction of an individual's genotype is determined, such as kuan katong genotyping by sequencing o katong basta.
Sequencing of human genome is already stored in GenBank. I
Oo.
hope you are familiar with NCBI GenBank, where you can find different DNA fragment sequences. So there are a lot of DNA sequences that you can
Napila yung
find
mga
there.
sources na nalimot
Yes, ang pinakakuan lang sa NCBI
ang
is
ipos mga site.
magamit jud siya nimo. I mean, ang NCBI manggod is open. There are other sites manggod
Oo,
nga
free.
restricted. Yes,
Dili.
restricted. And then
Okay.
Human Genome Project includes DNA sequences for human genomes. This is very important in the medical field, most especially.
Okay.
Okay,
So
go on.
let's go to the importance of the game. Has a vast range of uses across different areas, including ang agriculture, the forensics, medicines, o ang basic scientific research. So in agriculture, genotypic enhances the value of crops and herds. So understanding genetic variation has a particular benefit in the agricultural world as it enables the selection of traits. So, while ang traditional selective breeding involved purely katong observational methods, kanang selecting only plants or animals with superior phenotypic traits, such as kanang kinsay mas bugat or kinsay mas kusgan or kanang mas kuan for breeding. Ang modern selective breeding relies mainly or heavily sa molecular biology techniques, including nantong SNP genotyping. So, ang selective breeding pressures have generated breeds with more desirable phenotypes and changes to specific genomic regions associated with these phenotypes. So, in detecting these functionally relevant, as they say, genetic changes, helps us to determine kung which particular nga genes o ang sequences ang associated with sa particular nga phenotypic traits. So kanisha, it is useful for designing new and more intelligent breeding programs. So so with this use of kaning mga genotyping um, techniques, it has become a mainstay sa modern agricultural research na, like katong Illumina microarrays, which enable high throughput screening of kaning known markers o kaning array based studies, which was used in to discover novel associations between SNP markers and the traits or diseases. So let's go to forensics. So the importance of genotyping in forensics. For genotyping as basis in the characterization of forensic biological evidence for personal identification and kinship analysis. So personal identification and kinship analysis, like katong for example, paternity and maternity testing or DNA testing, which are the two main subject of forensic DNA analysis. Um, like biological specimens from which DNA is isolated, like motong blood, semen, saliva, tissues, bones, teeth, hairs can be used as a biological specimens. So ang genotyping has become a basis in the characterization of forensic biological evidence. So it can is performed using a variety of genetic markers. So in the case of medicine, importance of genotyping in medicine has a high predictive value for disease diagnosis and management o sa treatment. So genotyping can identify connections between SNPs and common disease risk by comparing the polymorphisms across two different populations. Like kanang one population could be healthy or one could be deceased. So genotyping can begin to unravel the biological processes underlying disease states by identifying the potential causal factors. So kaning genotyping or the SNP, it can also accelerate the personalized medicine by predicting an individual's risk of developing certain diseases or kanang designing targeted therapies specific to the genetic basis of the diseases. So as cunning SNPs are also as hunting SNPs also associated with individual therapeutic response. And also this the SNP based assays, it could also help select the best course of treatment. So as I have said, as you can see, genotyping in this context may help in controlling the spreading of pathogens by kanang tracing the origins of the outbreaks. Yeah, furthermore, naapod siya laing used in the transfusion medicine. For example, katong DNA based genotyping is being used as an alternative to serological antibody based methods to determine blood groups for matching donor to recipient instead of katong mga serological antibody-based methods. One could use DNA-based genotyping para to determine blood groups for matching donor to recipient.
Okay, how is it different with antibodies kaning sa DNA genotyping for determining different blood groups?
Mm.
Or what's the advantage of using DNA genotyping for determining blood groups before donation?
Kay murag mas dili siya gasto ay di siya cost ay
Hmm?
mas effective ang kaning transfusion medicine based on DNA genotyping kaysa serological anti-based methods.
I don't think so.
Ay, mas dali
Mas costly ang DNA genotyping because you have to use different
materials
chemicals.
technique.
Yes.
Ay, ko ano.
For example, primer is quite expensive.
Yes, ma'am.
Okay. But maybe in terms of accuracy,
Mas dali
mas
po dagway, ma'am.
accurate, no, kay ang
Ah,
sa serology, mas paspas ang kaning sa
sa
antibodies nga
antibody
paggamit.
methods.
Yes, mas dali man siya paggamit sa antibodies. Uh,
Yun yung gingon, ma'am, as an alternative round.
okay. But in terms of accuracy, maybe DNA
Uh,
genotyping is more accurate compared
accurate.
with the use of
Sige.
antibodies.
Yes, ma'am.
Okay.
So,
Okay.
last one is basic scientific importance of genotyping in basic scientific research. So, genotyping can provide detail and implication about an individual's evolutionary biology and history, as well as implications against future environmental challenges protection. So, kaning SNPs also have implications for evolutionary biology. So, genotyping can be useful in like identifying forms of genetic variation that underlies with kaning phenotypic differences between healthy individuals. So ang understanding this normal genetic variation across different populations would help us to understand how different groups have evolved and diverged, and also may have implications for protecting species against future environmental challenges. So also, genotyping applies to a broad range of individuals. So it includes microorganisms as well. So ang single-celled organisms such as bacteria also have SNPs. So ang SNPs genotyping can discriminate between bacterial isolates o ang between bacterial isolate, and can also be used to characterize strains of antibiotic resistance. Like for example, viruses o ang bacteria can be genotyped. So genotyping, uh, like I said, in this context, it can help in controlling the spreading of pathogens by tracing the origin of outbreaks. Yeah. Okay. So
Okay.
next,
Can you relate it to COVID-19 genotyping?
some of the genotyping methods, ma'am, are are used in making uh unsa ni in vaccines and kaning kanang unsa ni diagnosis for ay kaning PCR atong RT-PCR ma'am, like
Hmm?
like for example kanang making of saan to? Making of a vaccine.
In terms of transmission and efficiency in causing the disease, can you relate it to genotyping? There are some published articles now that are saying that genotype of individual also affects the rate of infection and production of or manifesting the disease.
Can saan kan
How is genotyping important in understanding how COVID-19 works on individuals? Or someone from the group can answer my question?
how
How about the others? Mr. Montegrejo?
Ay, sa individual ay sa host ma'am or sa virus? Sa host.
Host. Mm. Can you relate genotyping to the analysis or to how can you relate how genotyping can help in understanding transmission of COVID-19 or not really transmission but manifestation of the disease?
Okay. So, mo-try ra ko.
Mm.
The way the virus works is when it enters the body and and it binds into the receptors that is found in the cell. So, the receptors in the cell exist variedly. So there is variation in these receptors too. So if there is uh, a change in the receptor as the virus does, there must be some form of a lessening in the infectivity of the virus para siya maka-infect. Kay if the receptor or if the receptor nga naa sa virus changes because of certain mutations, it is also possible nga ang receptor nga naapod sa cell sa host could change as well. So if this receptor nga naa sa cell kay mo change pud siya, it is possible that there is a maka-confer siya og some sort of immunity against the virus.
So does that mean that if there is a change or mutation in the virus and DNA, does that mean that there will also be a corresponding change in the in
No,
the in the
no
host's
ma'am.
DNA?
No ma'am, no. Ang ako ipasabot is that if it is possible for the virus to have changes in its receptor, then it is also possible for the host to have changes in its receptor. Kay, it is sort of like an evolutionary arms race that if the virus
Mm,
changes, yeah,
okay. So if the virus changes, there will also be a possible change in the host level in order to combat the disease. Is that what you mean? Is that what you're trying
yes
to
ma'am,
say, Mr. Montegrejo? Okay,
yes, yes,
maybe, but not really totally possible. All right. How about at the individual level? I mean, at the human or the host level, in understanding how COVID-19 works in genotyping. Can you relate it? Answer? Can you answer my question? Did I give you the art? Did I give you the article last time on how COVID-19 affects different genotypes? Wala ko kahatan ninyo last time.
Wala, ma'am.
Okay. All right. So there is this theory right now that genotypes, different genotypes. Remember, although we belong to the same species, there is also a difference or genetic polymorphism in terms of our genotype, and this could be related to or what is the percentage or how is it possible that COVID-19 affects certain individuals. Okay, so they are trying to relate genotypes with efficiency of COVID-19 transmission. Okay. Say, for example, some races or human races are more affected with COVID-19 with severe symptoms compared with other races. So it's not really not just about the age, but could also be related to the genotype of the individual. Okay, that's in the case of COVID-19. Now, how about systematics? I know you have tried using molecular markers from NCBI to construct cladogram. So, can you discuss how genotypes are used or how is genotyping used in construction of cladograms for systematics? Ms. Elnas? Hello, Ms. Elnas?
Yes, ma'am.
Mm. Okay, so aside from systematics, this can be used in order to identify different variants of the virus. So, say sa COVID-19, so SNPs can also be used in order to identify different variants of uh, COVID-19. Okay, so in terms of systematics, molecular markers are used in order to what? So I suppose you understood why you need to use molecular markers sa systematics. Or did you try using si tawag ani DNA sequences belonging to the same species but from different geographical location? Have you tried that in systematics?
Wala, ma'am.
Okay. Sige. Sort of there is also study for that wherein they use the same species of organism, the same molecular marker, but they differ on geographical location. And they also found a significant difference among the molecular markers, which could actually be attributed to the geographical location. Okay, although it's not enough it's not this difference in the gene in the DNA sequence is not enough to identify this these organisms as another species, okay, or a separate species. Anyways, it's more on the systematic side. In case of evolution, genotyping can also be used to track evolutionary relationship among individuals or among species. And then, um, actually, in fact, there were some DNA sequences uh, that were used in order to identify evolutionary relationship among varying or polymorphic organisms. Okay, so although they differ in terms of their morphology, there was somehow a relationship to the on the DNA sequence of these organisms. Okay, I think. Okay, can you continue, Miss Elmas?
Okay. So next is the advantages and disadvantages of genotyping. So one of the advantages of genotyping um, is includes genotyping allows scientists to look at a much larger sample of the many nucleotide changes that differentiate one person's genome from another. And another advantages includes potential techniques could be universally available. So uh, next would be some of the genotyping techniques like ribotyping, pulsed field gel electrophoresis, and flagellant typing are already in use in a number of laboratories. And last one, since technology advances are not stationary, time and labor cost requirement of techniques can be alleviated. So one of the disadvantages of genotypings include is high number of untypable strains. Untypable meaning strains that are kanang dili wala pa siya categorized or assigned to a specific type or kind. And also genotyping can be time consuming. Kay several methods and processes are kanang undergone para like para ra ma-complete ang process. And also technically demanding requirements of techniques. So muna tong daghan kaayong kanang instruments o kanang uh, technologies ang gamiton. And also mahal pud lagmit ang mga kuan and ang mga instrument. And lastly, production and quality control of anti-serum reagents for serotyping schemes are costly and consequently, kaning mga reagents are not really widely available. So, dili siya for all.
Okay.
Yes.
Can you give relevant studies?
Ano sa ma'am?
Relevant studies.
Ah, irrelevant ma'am or biko related?
Yeah, yeah, related studies.
Ang sarap ma'am. So, one studies related to genotyping is the high throughput um, SNPs discovery and genotyping in hexaploid wheat. So, in this study, I kaning high throughput genotyping may be and um, means for like ang simultaneous genotyping for few to several hundreds or thousands of markers in hundreds or thousands of individuals. So, kuan siya like dako siya og coverage or simultaneously ang pag kuan genotyping. So, in this study, um, the TABW 280K was proven to be a very efficient tool for the diversity analysis as well as for breeding, as it can discriminate kung between closely related elite varieties. And also, the TABW 280K array was used to genotype a population derived from a cross between Chinese or Chinese Spring and Renan. leading sa construction leading to the construction of a dense genetic makeup comprising an 83,721 markers. So, kaning the results described in this study will provide the WIT community with powerful tools for both basic and applied research. Yes, that's all. Thank you.
Okay, mura to nga study
Yes, ma'am.
related.
Mura akong
Because
nakuan.
there are a lot of
Gani,
studies
mura akong
that uses genotyping as their main
created.
application in order to discern certain species or certain strains. This is widely used in agriculture. Muna ni siya gamit ron sa agriculture. So wala na tong mga old
Mas
ways.
efficient
Although
man.
if
I mean,
efficient man gihapon siya in terms of farmers, even Filipino farmers are still using that method in which magkuan sila, mag-cross sila and to test which variety is more efficient in production of fruits. For example, sa tomato, even local farmers in Balinsa Sayaw still use that traditional method. Sila ray mag-breed to find out unsay pinakamaayong variety ang angay itanom sa ilang lugar. Okay. Although ang trend karon is to use um, the genetic sequence or DNA sequence in order to identify varieties nga mas makaproduce of fruits in case of um, mga plants nila. And then in the case of kaning sa ilahang animal nga side, they use genetic markers as well to identify sani, uh, individuals of these meat producing organisms to find out asa ang mas makagama o bugat nga meat and then mas efficient in production of milk para sa ilahang dairy products. Okay. Anyway, that's for agriculture. I, it's so much better. Maybe you can add other studies as for example sa katong imuhang mga example Miss Elna sa forensics
yes,
and
ma'am.
then evolution next time, next meeting, maybe you can add that one.
Okay,
So,
ma'am.
bisag isa ka article lang,
Okay.
representative for each application.
Okay, ma'am.
Okay, thank
Yes,
you.
ma'am.
Next presenter.
So good morning, everyone. Um, so my topic is about the next generation sequencing. So this is one of the different methods for in sequencing. So the outline would be introduction to the NGS applications, and a detailed next generation sequencing methods, which includes the library preparation, cluster generation, sequencing, and data analysis. So the next generation sequencing, or NGS, also known as massively parallel sequencing, has completely revolutionized genome analysis. Um, with with the next gen sequencing, it is now possible to sequence an entire human genome in just 1 day. So with its ultra high throughputs, scalability, and speed, NGS enables researchers to perform a wide variety of applications and study biological systems at a level that is never done before. So the innovative sample preparations and data analysis options enables a broad range of applications. For example, next-gen sequencing allows researchers to rapidly sequence whole genomes within a short period of time. This is relative to the old methods, and it deeply sequences target regions. It sequences cancer samples to study rare somatic variants, tumor subclones, etc. And it studied the human microbiome, and it identifies novel pathogens. So the next gen sequencing is similar to CE sequencing, and it's the critical difference of the two is that um, instead of sequencing a single DNA fragment, which consumes a lot of time or even several years to finish, um, the NGS extends this process across millions of fragments in a massively parallel fashion, which um, leads to a significant increase of speed when um, when sequencing. So the Illumina is a company responsible for more than 90% of the world's genome sequencing data through sequencing by synthesis, or SBS, with which delivers high accuracy, high yield of error-free reads, and a high percentage of base calls. So these are the steps. Are the Illumina NGS workflows includes four basic steps, which I included some um, simpler diagrams. So first step would be the library preparation. So the sequencing library is prepared by a random fragmentation of the DNA and a complementary DNA sample, as you can see in the picture, in this figure. So the which is followed by a 5' and a 3' adapter ligation. So fragmentation is termed, um, which combines the fragmentation and ligation reactions into a single step that greatly increases the efficiency of the library preparation process. So um, first, in the library preparation, the genomic DNA is isolated, and then fragmentation of this DNA will take place to generate smaller fragments. So these DNA fragments will be joined and then ligated to certain oligonucleotide, or by 5' and 3' adapter ligation. So to summarize, the genomic DNA is in fragments, and all these fragments have attached two different types of binding sites at each ends. So after that, it binds to the flow cell. So all the DNA fragments that are now applied to the chip, which termed as the flow cell, and as you can see on the simpler example on the left, um, there are two types of complementary sequences, which are itong 3 prime and the 5 prime, which are in colored red and blue, and the DNA fragments will then hybridize and with the flow cell, and which is on the plate. So after ato, it undergoes the process called DNA amplification by PCR or polymerase chain reaction. So the first step of the flow cell in the flow cell is the DNA amplification by PCR or polymerase chain reaction. The complementary sequence to our signal strand kaning first na naa sa flow cell kay ang atong signal strand, and then it undergoes a PCR, and the complementary sequence to our signal strand DNA fragment is synthesized. This process is done simultaneously to all DNA fragments of the whole flow cell. So dili ra siya usa kay kuha na siya para ma-focus ra and for simplicity. So muna siya kaning nasa right mo siya ang kuan kanang image sa flow cell. So the DNA undergoes denaturation, and the signal strand, which is not attached to the flow cell's oligonucleotides, gets washed away, and the remaining complementary DNA strands undergoes bridge amplification. So as you can see here, after to ni ni undergo og DNA amplification, katong wala na katong strand na wala ni attach sa flow cells oligonucleotides kay na kuan na washed away after undergoing denaturation. So so um so so as you can see, that we have two different oligonucleotides here, and the second type of oligonucleotide will now hybridize to one of the sticky oligonucleotides on the plate. So sila na duwan nga ni stick na siya sa oligonucleotides within the katong ni attach sa flow cell. And this process is what we call a bridge building, and it again undergoes DNA amplification, but still with through PCR or polymerase chain reaction to synthesize a complementary strand.
Why is DNA amplification needed for DNA sequencing?
Ay, sorry. Fun. DNA amplification is needed sa sa ani na process kay para maka generate from um, one DNA fragment, para maka-generate siya og millions or thousands of copy ana na DNA na sequence mo.
Okay, so why is it important in DNA sequencing? DNA amplification using PCR. Main purpose is to create multiple copies of that particular DNA. But why does it need multiple copies before DNA sequencing? Hello?
Yes, ma'am.
Mm. Okay. Can someone answer my question? Why is DNA amplification important for DNA sequencing?
Um, I think, kuwan, ma'am, its purpose is para to make enough copy of the um, targeted na DNA sequence, ma'am, or particular na genes.
Okay. And aside from that, in the statistical context, okay, it's also important to create large samples of DNA for verification purposes and for statistical purposes. That's why before DNA sequencing, this DNA molecule must be amplified first and create multiple copies before subjecting to sequence. Okay, continue, Mr. Gador.
Um, so um, so this process will be repeated for several rounds until it generates multiple copies of multiple copies of the single genomic DNA fragment. So this process will so we now have the reverse and forward strands from our copy, and then reverse strands will be cleaved, and then sequencing will be done next. So in cluster generation is the second step of the process. So cluster generation for cluster generation, the library is loaded into a flow cell where fragments are captured in a loan of surface-bound oligos complementary to the library adapters. So each fragments are then amplified into distinct cloner clusters through bridge amplification katong kaganina, and when cluster generation is complete, the templates ready for are now ready for sequencing. So and focusing sequencing, the Illumina SBS technology uses proprietary reversible terminator based method that detects single bases as they are incorporated into DNA template strands. Um, and focusing uh, only on one strand, as mentioned earlier, a primer combined to the oligopeptide sequencing binding site now sequence sequencing is done with specific nucleotide. So these nucleotides are fluorescently labeled and only one of them will exactly fit. So as you can see here, the fluorescently labeled nucleotides, example kaning T, the thymine is in colored green, the A is red and yellow ang G o blue ang C. So the first would be kaning fluorescently labeled na T in green will be excited by a laser of the fluorescent signal when fluorescent signal is obtained. So after that, muna na, ang kanina copy will be the one um, will be recorded. So muna siya ning na diri. T A A C G T. And so because of the PCR or the polymerase chain reaction, we had several copies and many light signals are obtained simultaneously. So muna siyang fast and efficient ang kaning NGS kay tungod atong kay simultaneous man siya nga nagdungan og read sa signals and
Go on, Mr. Gador.
kay bisag daghan kay siya og copy maabot tayo og millions, they are obtained simultaneously and recorded. And the last step is the data analysis. So this would be the last step, and the data analysis and alignment. The newly identified sequence reads are aligned to the reference genome. In this stage, informatic tools are required. So following alignment, many variations of analysis are possible, such as single nucleotide polymorphism, katong gidiscuss ni Elnas kaganina, and or insertion, deletion, identification, and all and others dayong katong iyang benefits, which are also discussed kaganina. So mura to siya taman ang methods sa NGS, and after sa ma-analyze na ang data mo undergo na siya og variations of analysis for different purposes.
Okay, that's in the case of genotyping. Okay, that is if there is a reference genome. However, if there is none, then it can only be used for genetic sequencing. Okay, so what are the advantages and disadvantages of this particular method? Did you include it in your report, Mr. Gador?
No, ma'am.
Okay, next time, next meeting, please include advantage and disadvantage of NGS, and then I'll also give different related articles or relevant articles in which NGS is used as one of the methods for genetic sequencing. Okay,
Okay, ma'am.
all right. Next reporter.
Can I have a moment? Can you log akong laptop?
Okay. We'll wait for you, Mr. Montegrejo. I think Banagudos and Mr. Plit cannot report this morning, so we can have you guys next week Tuesday. And then Mr. Salapa, okay, yay ka din na Banagudos. Mr. Salapa,
Yes, ma'am.
ikaw may present sa article ha. I will post
Uh,
the article. Yes, I will post the article for your critique maybe this afternoon.
okay, ma'am.
Sa Google Classroom, rin ako siya i-attach, and then deadline will be probably Let me check. Deadline will be on Tuesday, April 20. April 20 ang deadline.
Kanus
Unya,
ano ka
guys, i-present nimo siya after nasa presentations tanan. So, maybe dili pa kaka-present this coming Tuesday, pero prepare lang. Ay, Tuesday. Ah, yes, Tuesday. Prepare lang, basig maka-present ka. Basta, kuan na lang. Ay,
yes.
dili, di, April 20 ang deadline, guys. April 22, 1 week. Tagan tamog 1 week. So, 15 man ako i-post. Karong 22 ang deadline sa critic paper. 20 mo present pa si Banagudos o si Triplet. Hasta si Gador o Elnas ato inyong kulang. Okay. 22 na tamag koan. Unya, is it okay if the same lang gihapon rule sa microbiology before? Ang atong quiz is every Saturday na lang morning. Walay conflict sa inyong schedule? Saturday morning? Wala ra.
Wala ra, ma'am.
Okay. Huwag kaya pumilod sleep. Di ba natagaan mo? Pero kabalo mo sa inyong
Wala
subject.
Japan, ma'am.
Pero
Wala,
kabalo mo sa
ma'am.
inyong subjects.
Wala, ma'am.
Imo sulyara ugma ra na namo ko anon kay klase ko tibok adlaw ron.
Kaya, ma'am.
As for the others, pero nakasulod na mo sugod. How about mga second year people nakasulod na mo sa inyong minors?
Yes, ma'am.
Okay. Wa pa may klase sa chem? Or naa na mo
O sa
instructor?
biophysics, good.
Wala pa may instructor, ma'am.
Wala pa gihapon. Amo niya na ikuan. Unsa naman niya eh? Amo niya na i-check. Ano yung biophysics ninyo? Murag inyong biophysics
Yeah.
engineer man siguro may mo handle ana inay.
Na naman tayo nangayo o kuan, ma'am.
Wala pa na
Nangayo naman
kuan
kung
ng
details si
fix.
bedroom na mo.
Mm. Hulat-hulat lang sa mo siguro pa sila na magbahin-bahin sa load. So, ato ang quiz every Saturday, 10 or 9. Ito yung preferred time.
Pwede 9 ra, ma'am.
By the way, sa inyo nga quiz, kuan sa ta? Written. Okay. 9. Yes. Ah, sige, written sa taon niya na ako mag-oral. So, we'll start at 10 a.m. for your written exam. Maybe next week, Saturday. Dili pa karong Saturday. Okay na, Montegreho? Hang gyapon?
Itikig siya, ma'am.
Tikig siya. Ari ta mag-unang Banagudos. Tikig pa ang kang Montegreho. Apa may time? Yes, makita ra. Mr. Banagudos, before you continue, what does C stands for cDNA? Okay, go on. Beto galibog po ko nimo Mr. Banagudos, pero akong question Mr. Banagudos is why is RNA extracted and used for identification of the sequence, DNA sequence? What is use? What is the process? What is the okay? So why is it RNA man ang iyang gigamit? And not just ay nan dili man diretso DNA. Why is it RNA man ang gigamit niya? Instead of just using DNA, since RNA will still undergo reverse transcription to produce complementary DNA. Diba? DNA man siya po niyang gamiton. Gamit mo man siya RNA. Gadugang-dugang steps or naay purpose ano. Based on what you have learned in protein synthesis. Anyone from the group who wants to answer the question? Nganong RNA ang gigamit instead of just using DNA directly? Tubil, Tubil, are you with us?
Yes, ma'am.
Okay, kindly answer the question. Oh, Banagodos.
For
I think Banagodos is ready. Ah, sige, sige, sige. Ikaw sa Tubil. Then we'll go to Banagodos.
I I maybe ra ni ma'am nga like sa maybe they use RN RNA kay katong sa eukaryote because I think eukaryote um uh kay 'di ba sa mga eukaryotes ma'am kay kuana sila introns so dili siya so without this RNA dili siya ma-splice dili siya ma-splice so maybe that's the reason why kailangan pa siyang RNA usa i-kuan for to generate DNA.
Are you sure?
Dili, ma'am.
Okay. Mr. Banagodos, maybe you can continue first and then we'll go back to my question after you explain the steps. Unsa to? If fluorescent ang sa tumor o sa normal dayon, sige go on. Okay. Second question, Mr. Banagodos. How did they know that genes were inactive in both groups? Kanang naka-black. Okay. Therefore, kindly relate it why they used RNA. Why they use RNA? Actually, you're there. You have already explained. But I just want to know if I just want to check if you know why they are using RNA instead of just directly using DNA. Since they still have to convert it back to complementary DNA anyway. Mm. Okay, that's correct. Remember, according to central dogma of molecular genetics, DNA will be transcribed into mRNA. So you will know that the gene is expressed once it's converted or transcribed into mRNA. Okay? And then, next question, nganong i-convert man siya back into complementary DNA? genes were not expressed since they are using mRNA man. Okay, and then undergoes reverse transcription to produce complementary DNA. Okay, so based on the result, what does that red? I sorry. So based on the result, what does that gene colored red mean? Kanang reds sa letter C nga dear Mr. Banagudos. What does it indicate? Mm. So, what does it indicate? These genes are expressed maybe because of treatment. Okay? Whereas kanang green are suppressed ang iyahang expression due to maybe treatment. Okay? How about yellow? Or it may indicate that these genes are not affected by the treatment. Why? Because they are equally active in both treated and untreated cells. Diba? Mm-mm. So, so what does it mean? Okay, that's correct. Ang iyahang expression also varies. Okay, next. Okay, go on, Mr. Banagudos. Unsa man? Efficiency? Okay. Can you compare microarrays, DNA microarrays with the report of Mr. Gador, NGS, based on their application? Unsa man siya mas efficient? Why? Fragments. But in terms of okay, so that's that disadvantage. Okay, how about advantage NIA over NGS in terms of application? Hmm. Okay, can you answer the question, Mr. Gador? What do you think is the advantage of using DNA microarrays, microchip over NGS?
Mas specific dagay ang kanaya
So to, Mr. Gador, mas specific?
wait sa
Okay. Okay, so in terms of gene expression, um, mas ma-detect sa DNA microarrays on what are the genes that are expressed at a certain time. Okay, so unlike sa NGS that it just analyzes the DNA sequence in using microarray, DNA microarray can detect the genes that are expressed by using RNA. Okay, although it could also be used in NGS, can also be used in NGS by using RNA and then converted back into uh, complementary DNA before CIE sequence. Okay. Do you have report pa to follow, Mr. Banagodos, aside from this? Why? That is sample size issue, I guess. Okay. In case of development, Mr. Banagudos, unsay ang application? Development of organism, like development of eyes, development of the organs. Okay. So some cells can be used at an early stage of development to check what are the genes responsible for the development of certain organs. Okay. So for example, at the early stage of the development, ilahang gamiton ang unsa ka part sa organism or early stage sa development, gestation period, ilahang gamiton ang iyang DNA to check o iyang RNA to check what are the genes expressed and what are they responsible for. Okay. So aside from this, as a development and the medical aspect, what else? Sa pa application sa microarray. Okay. From sa SNPs. Okay. Alright. That's all? Okay. Thank you, Mr. Banagodos. So we will continue with our report or with the report of Mr. Dador and Miss Elnas next week Tuesday, and then followed by Mr. Montegrejo. So paunahon sa nato si Miss Elnas i-present sa atong uban nga articles, and then followed by Mr. Gador, and then usa ka Mr. Montegrejo. Okay?
Yes.
Yeah. Question, Mr. Bana. Asa pasabot asa? I'm not really sure with highly reproducible. Not really sure with that, Mr. Banagodos. Maybe iyahang kuan. Imong giwala, nabasa pa gani ko. Imong gi and nabasa pa gani ko. Ibalik sa to Banagodos. Iyaha sigurong efficiency sa pagkuan. Not really sure. Let me read on that.
Kuan, ma'am. Reproducible means can be kanang kuan ba murag sa research gani kanang ma-study nimo siya again with the same methodology pero on a higher degree.
So example, so pwede pa siyang gamitog balik.
Mm.
Okay.
Ano, excuse me. Iway na.
Okay. So unsa nga mga example ang law ang iyahang kuan, Mr. Banagodos? Ato imong giingon ba nga ang uban is highly reproducible and others are not? Unsa nga example? Gani. So nganong highly man siya reproducible ang oligonucleotide? but I am not really sure. Let me check on that. Okay, so mas less ang sa spotted microarray compared sa oligonucleotide. Maybe there is this particular step siguro sa oligonucleotide microarray that makes it highly reproducible. Let's read on that. Let's read more on that. Okay, thank you. Alright, sige, stop na. Wala na question before we end this call? Okay, wala na. Thank you for attending our class. You may now leave this call. Stay safe. Bye.
Thank you, ma'am.
Thank you, ma'am.
Bye.
Thank you,
Thank
ma'am.
you, ma'am. Goodbye, ma'am.
Bye.
Thank you, ma'am. Goodbye, ma'am.
Mauna ta. Huwag lami ang sudan.